Welcome everyone to our lecture today. It's a real pleasure to have Professor Claudia Höbartner with us from the University of Würzburg. And she's the winner of the Family Hansen Award 2023 in Medical Sciences. And she will give a lecture about enzymes made of RNA or DNA. And before we dive into the topic with Claudia, I would like to introduce you shortly to the Bayer Foundation for those of you who might not so be familiar with the Bayer Foundation or have never heard of us. So what is it all about? And we are actually hosting this event today. The Bayer Foundation, we are working at the intersection of health, nutrition, and environment. And if you want, we are the philanthropic branch of Bayer. And we are looking into, on one side, into social innovation and how we can here actually improve communities and foster social entrepreneurs in the global south. And on the other side, the other pillar we are focusing on is the science part. And here we are looking into how we can advance science and promote science further. And that's actually the two pillars we are concentrating on. And diving a bit more into the scientific arena of the Bayer Foundation. So here we focus on advancing science on one part, and then on the other side also on equity and science. And here we want to increase the equity in science by building scientific capacity in Africa, mainly in sub-Saharan Africa. And we have here different collaborations and support a research hub, for example, but also postdoctoral fellows that they can pursue their research in Germany and German institutes. And also fostering gender equality is another important topic for us, where we support female scientists in their career development. And then advancing science, the other pillar, we honor outstanding scientists for their scientific research achievements and as role models. And that's where Claudia Höbartner actually received the award for the Hansen family. And that's why we are having her here today. Another important item or topic for us is to enhance trust in science, and here really start with the next generation of future scientists. So here by fostering and supporting STEM education. And what is also very important for us is to foster interdisciplinary exchange, as we have it summarized here on the slide. This is actually that we have fellowships where PhD students, master students, medical science students can apply for. And also that we have our lecture series and symposia, which are summarized on that slide. So we have these scientific lectures, or boundary-breaking science lectures, as we call them, and also a symposium organized together with our Bayer Science Fellows. It just took place in April this year and was focusing on artificial intelligence. And all of these sessions are made available via our YouTube channel of the Bayer Foundation. And it's available for our R&D community internally, but also for our scientific network outside of the company. And with that, some housekeeping. Just to remind you, this session is being recorded. And if you have questions, you can post them in the Q&A function, which you should see on your screen below. And also we have later on a discussion where you will be able to open your microphone and your camera, and I will give you further details on that later on. So you can also ask your questions right away, then later after Claudia has finished her lecture. And with that, I will hand over and again give a warm welcome to Claudia Höbartner, our Hansen Family Award winner 2023. And looking forward to learn more about her research, what she has been working on, and then looking forward to all your questions. Thank you. Handing over to you, Claudia.
Thank you very much. Good afternoon to everybody. It's a great pleasure to be here today. Thank you very much for organizing this great opportunity to share something about our research. I have been fascinated by nucleic acids since ever I started studying. And my research group now focuses on many different aspects of RNA and DNA, synthetic aspects, but also we try to understand biology. And I would like to share some of the questions today with you that we address in the lab and some of the results that we recently found. And in order to do so, I would now like to start sharing my screen. And I think that should be functioning in a second. So.
Yeah.
Yes. I came into studying the nucleic acids by being fascinated how life is possible. We know that all the information that a living cell needs is stored in the DNA. And in order for the proteins to be produced, the information first is transcribed into RNA before it is translated into proteins. And when we look at different types of an organism, we see very different types of cells with very different functions. The functions are mostly defined by the proteins that are expressed at a certain point in time. But it is quite interesting that those two different cell types of the same organism share the same information, although they make different proteins. In between is the level of RNA. And we now know that about 80% of the information stored in DNA is actually transcribed into RNA. But only a very tiny fraction of 3 to 5% is actually translated into proteins. So that means that in this alphabet of RNA, there is so much more information stored and additional functions that these RNAs make that we are not fully understanding yet. One reason for that is that we have very limited tools available that allow us to study RNA, to visualize it. So when we look at these types of fluorescent pictures, mostly we have tools available that allow us to visualize proteins. The tools available for studying RNA are much more limited. And this is one of the motivations and directions of where my research group tries to contribute. We would like to develop new nucleic acid-based tools that allow us to study RNA chemical biology and also then allow us to develop new therapeutic and diagnostic agents. The large diversity of functions that RNA has also has to do with the fact that it not only uses those 4 standard letters of the genetic code, but in addition, these 4 letters are decorated with chemical modifications at all these positions that are marked here with these arrows. There is now about 150 different modifications known in nature, and they influence the structures, but also the folding states, interactions with proteins, lifetimes of RNA. All levels of gene expression are somehow influenced by these modification states. And these modifications are introduced by protein enzymes, which are referred to as writer enzymes. And they are recognized and their function is translated by reader proteins. Some of these modifications can also be removed by enzymes called eraser proteins. These types of modifications or small chemical changes on those nucleobases, they also play a role in synthetic nucleic acids. For example, in those RNA-based medicines that we have learned a lot in the recent years. For example, mRNA-based vaccines have tremendously benefited from the change of a standard uridine nucleotide into this methylated pseudouridine nucleotide. And also small, short antisense oligonucleotides that are used for regulating the gene expression of certain mRNAs, they also are modified either at the phosphate backbone or at the ribose with those tiny chemical modifications. So one of our interests is indeed in understanding the natural functions of RNA modifications, but also to help develop new modifications to enhance functions and applications. So beyond the function of transferring or helping the translation of the information into the proteins, three-dimensional structures of RNAs also play a very important role for their functions. And that's why we often refer to them also as functional nucleic acids. And here I highlight two classes of additional functions or of types of functional RNAs that we also study in my group. And these are, on the one hand, aptamers. You can consider aptamers as the nucleic acid equivalent of an antibody, something that can recognize a target or bind a ligand, so in a non-covalent interaction. For example, small molecules that would not be fluorescent in solution could be bound to an RNA, and this binding event could light up the RNA. In this way, we can develop tools that allow us to visualize RNA. On the other hand, the second type of functional nucleic acids that we are very much interested in is those that can catalyze chemical reactions, and they are referred to as ribozymes. And that is what I will spend the major part of this talk today. These are enzymes that are made out of RNA or DNA. And one direction in the context of how to visualize RNA will be to develop ribozymes that can install a fluorophore, a fluorescent label on an RNA, or a cross-linking agent that can allow us to study RNA-protein interactions, or a ribozyme that could simply install one of these natural chemical modifications. So in general, you can say my lab tries to combine those two areas of research, RNA modifications, with enzymes made of RNA and DNA. And more generally, you can say we try or we ask the question if RNA and DNA enzymes can mimic the functions of protein enzymes to write and read and potentially in the future even erase RNA modifications. This is what is summarized on this slide as these two general directions of RNA. But before I show you a small story on each of these two topics, I'd like to give a brief background on nucleic acids catalysts in general. So when we talk about ribozymes, we all mostly talk about those natural RNA enzymes that were discovered by Tom Jack and Sidney Oldman in the beginning of the 1980s. For this discovery, they were awarded with the Nobel Prize. They found that these are self-splicing RNAs, or RNAs that are processed by themselves during the maturation of the RNA. And they catalyze transesterification reactions, cleavage and ligation events in the RNA backbone. Also, small classes of nucleolytic ribozymes have been identified that catalyze this transesterification via the attack of this 2' hydroxy group on the neighboring phosphate atom. And these enzymes, in principle, are structurally and mechanistically pretty well characterized. But it's still a highly vibrant area of research, which is reflected in several recently appeared papers that have shown that these ribozymes even play roles in the human genome. And very recently, even a new class of group 2 intron ribozymes has been described that catalyze the hydrolytic endonucleolytic cleavage of DNA. And I think this can immediately remind you on what we all know what CRISPR is doing. Namely, we have tools that can target DNA to reprogram or re-engineer gene expression pathways. And in this context here, we would now be able to do this like projecting into the future just with a piece of RNA that can manipulate the genomic information of a cell. So ribozymes have been found in nature, are studied a lot, but can also be generated in the laboratory. Ribozymes as a class of enzymes that are made of RNA. So now we can ask the question, what about enzymes made of DNA? They would be called deoxyribozymes. And these types of enzymes have so far not been found in nature. We can speculate for reasons about that, and I'm happy to discuss about this later if there is maybe the potential to eventually find them out there. But so far, DNA that has catalytic activity is only generated in the laboratory. And you can also imagine sort of what are the requirements for a DNA to fold into a tertiary structure that can catalyze a chemical reaction. Obviously, it needs to get released from the stiff double-stranded duplex DNA state that we would normally know. And so DNA enzymes have also been studied now since long time. The first one was reported in 1994. And most of the known DNA enzymes also are very good at catalyzing phosphotransfer reactions of the similar type that ribozymes catalyze, the cleavage and also ligation of RNA backbones. And so how would one now find such ribozymes, new ribozymes for other types of reactions or for these RNA cleavage and ligation reactions in the laboratory? The procedure that is used for that is often referred to as in vitro selection or also in vitro evolution. And it means that it starts from a synthetic library of DNA or RNA with a randomized region that contains at each position with equal probability one of the 4 nucleotides. So quite often we would use 40 nucleotides, or in principle, we're not limited to whatever we think could be a good number to form an active site eventually. But even for only 40 nucleotides, there is 4 to the power of 40 possible sequences. But this is much more than we can examine in the laboratory in a reasonable experiment. So usually we would only have in our experiment roughly 10 to the 14th different sequences. And from these is still a huge number that we cannot examine in parallel, like we would do in a screening approach, where each of our wells or our sample vials would contain one candidate sequence. This is what we would call a screening approach. In a selection approach, in contrast, all these 10 to the 14th are combined in one single tube. And by repeated cycles of selection and amplification, we enrich those that catalyze a particular reaction. And then after several amplification rounds, we will see enrichment, we will start cloning and sequencing, and then have an individual DNA sequence, defined DNA sequence, 40 letters after each other, that then falls into a 3-dimensional structure, which we also would like to understand and analyze, and then can be developed into a tool. So when we now start first looking at the question, can a DNA enzyme distinguish modified from unmodified RNA? So that would mean it mimics a reader protein, it recognizes the modified RNA and then decides, well, okay, this RNA is modified, so I should not degrade it. Or opposite direction would also be possible, that the modified RNA is preferentially degraded and the unmodified RNA is kept intact. So in other words, what we would be looking for is a DNA enzyme, deoxyribozyme, that catalyzes this transesterification, but it distinguishes, it makes, it checks if this adenine, for example, carries a methylation, either at its exocyclic position or at the internal position. And in ideal case scenario, it will not only check for the presence of the modification, but it would also be able to distinguish these 2 different modified isomers of this methylated adenosine. So as I mentioned, the first or the most well-studied DNA enzymes are exactly those that catalyze this reaction. And some of you may have heard about 1023 or 817 DNA enzymes. These are really the most widely used in any biochemistry or synthetic nucleic acid chemistry laboratory that uses DNA enzymes to cut RNA at a particular site. And so these DNA enzymes, they were never trained to look at a modification. So we were first asking the question, would it actually make a difference if this adenosine is methylated or not? So we had 2 RNAs prepared, one carries the modification, the others not, and then look at the cleavage yield over time, and you see basically there is no difference. So that means if we want to use a DNA enzyme to distinguish those 2, we need to train it. And that training process is exactly what in vitro selection is about. This is the rough scheme how this is done. We have our DNA library. It is connected to the RNA substrate, which carries at this red dot the modification, either the methylated or the unmethylated nucleotide. And then in the selection step, we separate those molecules that are cleaved from those that are non-cleaved. And those that are cleaved would be active, can be amplified, again attached to the substrate and gone through the next round of selection. And if we play this in the forward direction and alternate modified and unmodified RNA, we can train the DNA enzymes that we obtained from this experiment to preferentially cleave M6A modified RNA. But you can well imagine that we can also play this game in the opposite direction and enrich RNAs that preferentially cleave unmodified RNA. And those would then be inhibited by the modification. And indeed, for both types of questions, we found DNA enzymes that can do that. Here is just one example of a DNA enzyme that is strongly inhibited. Now, earlier you saw that both curves were overlapping. Now we have a really huge difference. We have 50 to 150-fold faster cleavage of unmodified RNA and strong inhibition by the presence of this modification. We showed that these can even be used for natural RNAs, such as these CD box RNAs, to check the modification level of those transcripts. We played this then for many different families of RNA modifications. For example, cytidine modifications are an extremely interesting class of modified nucleotides. We have several different isomers. Some of them are shown here, where the methyl group is either positioned at this ring nitrogen here, which generates a positive charge, or at this exocyclic amino group. There is no positive charge, and there is two orientations possible, syn or anti-orientation of this methyl group, or the methyl group can be attached to the C5 carbon. This is a very important also epigenetic modification that is also found in DNA, and this is important there for switching on and off transcription of genes. In the RNA, it's also found in many kinds of RNA. It can be even further modified, where this methyl group then is oxidized to hydroxymethyl formyl groups. And this is also an area that we have studied a lot. Again, very similar procedures. We find catalysts that can specific for each of these modified nucleotides. And you can imagine that this gives us quite a number of different sequences to be studied. So we take advantage of high throughput sequencing analysis to generate activity profiles for thousands of deoxyribozymes. And to do so, we basically sequence cleaved and uncleaved RNA in one go, and can then generate these profiles where we compare the activity against modified RNA to the activity against unmodified RNA. And so here is now one example shown for this M3C, 3-methylcytidine. So any dot in this plot corresponds to one specific DNA sequence. And any dot in this upper left corner here shows us DNA enzymes that are activated by the presence of the modification, whereas those down here in the lower right corner would be candidates that are inhibited by the modification. And the color code represents what is the nucleotide that precedes this modification here, this N. So you can see that there is really a large number of variants, and we have now, with this type of analysis, a direct prediction of the selectivity. And we have tested several of those then experimentally and showed a very good correlation between this activity profile suggested by the sequencing data and the in fact RNA cleavage activity. So we think that this method allows us to accelerate the discovery of modification-specific DNA enzymes significantly. And so why am I now interested in this, studying these modifications by DNA enzymes? And that brings us to a very interesting example of crosstalk of modifications in the anticodon loop of mitochondrial tRNAs. Mitochondrial tRNAs are quite heavily modified, like all tRNAs are quite heavily modified. And some of these modifications, the presence of one modification may be necessary for the other to be installed. And if there is a defect in one of these enzymes or a lack of one modification, the downstream effects may be affected. The downstream functions of the tRNA may be affected, so that a particular protein is not expressed anymore properly because the tRNA is not properly folded and cannot be used by the ribosome in the way it should. And in order to examine the modification state of natural tRNAs that we get from natural sources, cell cultures, for example, or directly isolated from mitochondria, we can then use these DNA enzymes to examine was the modification present or not at a particular state or in a particular cell line that reflects a disease model, for example. And for this, we teamed up with Markus Bonsak's lab in Göttingen, who studies those enzymes. He's in the medical department and studies modification enzymes and also disease-related modifications. And with our tools, we could now really demonstrate that we can examine the modification state in a very specific manner. So there is way more to do with these DNA enzymes. What I showed you so far is how we find them, that we have now pretty good tools for a quantitative assessment of the modification level at the particular target site, and that these deep sequencing-based approaches really allow us to profile large numbers of RNA-cleaving DNA enzymes. And so forward-looking, we think that we can develop these tools for many of those relevant RNA modifications in nature. So the second part I'd like to show briefly is addressing the question of now finding ribozymes that can install a modification. This sort of expands, goes now away from the manipulation of the phosphodiester backbone, but asks the question, what additional functions, reactions can RNA catalyze? And so in particularly, it has been a very long-standing question if a single methyl group can be introduced into an RNA by a ribozyme. And this is also related somehow to the question of RNA evolution, to the RNA world theory, when we expected that RNA has additional functions, or that RNA by itself could replicate and store genetic information fulfills both of those functions. Also, these methylated nucleotides are pretty ancient in evolution. So there has been long the hypothesis that maybe RNA could have catalyzed these types of modifications or could have also repair functions early on in time. And so it has really been a long-standing question to find such ribozymes or to reconstitute such potentially lost activities of RNA in the laboratory. And you can imagine that this is sort of challenging to find a ribozyme that does this tiny little modification. Earlier, I showed you we separate based on cleaving the RNA backbone. So there's a huge change in molecular weight. We can easily separate by physical properties. But attaching such a small methyl group is really challenging. So we need sort of a proxy for the methyl group. And that is, so one trick that is generally used in the field is to use a larger alkyl group that in reactivity-wise is very comparable to the methyl group, but then has an affinity handle on which enrichment is possible. And the most often used affinity handle is biotin that can be enriched by streptavidin chromatography. But what would be now a good methyl group donor? And here we took inspiration from nature, from methyl transferase enzymes, but actually from a DNA repair protein. So many of you may also be familiar with the SNAP-tag. The SNAP-tag is a protein labeling tool that was developed by Kai Jonsen's lab. Kai Jonsen was the previous recipient of the Hansen Family Award. And he was also inspired by this methyltransferase enzyme that alkylates itself by taking away this methyl group from a DNA onto the enzyme. And he engineered this enzyme in a way that it can now use not a full DNA strand, but only this guanine that carries this benzyl group. And whatever is then attached here at the end of this benzyl group would get attached to the protein. And this allows then for fluorescent labeling or for attachment of cross-linkers onto the protein of interest. So we took this idea and thought that we can replace the protein enzyme here now with a ribozyme. And in this way, we would generate an alkyl transferase ribozyme that benzylates itself, and then see if we can sort of reverse engineer it into a methyltransferase. The in vitro selection scheme is sort of similar to what I showed you before. We have this library where we have the modification site on where we think the modification should happen. We enrich those reacted RNAs by this streptavidin binding, amplify it. First need to go back to DNA, then amplify the DNA, transcribe it again into RNA, and go through a couple of selection rounds, and then identify individual sequences that we can study. Here is such an example. A particular sequence with 40 nucleotides that comes out of the selection that was done with this benzylated guanines. And you can see when we incubate the substrate RNA, this is this band here, with the cofactor and the ribozyme over time, a new product is formed. And the key question now was, what if we provide not the benzyl group with the biotin, but only the methyl group? And we also see a new product being formed. And this was super exciting, but I was really skeptical at the time because I was not really expecting that I can see a shift of a single methyl group on an RNA, which is only 14 mass units. So really confirmation then came by doing mass spec analysis and showing that indeed the RNA has gained 14 mass units. So the key question was now to figure out where the modification sits. And this story I really have to cut short. I can tell you what we found is our first methyltransferase ribozyme really catalyzes the installation of one methyl adenosine in RNA. And it uses this small molecule cofactor 06 methylguanine as the donor of the methyl group. We characterized this in detail regarding reaction conditions, also sequence dependence, and could show that indeed, this ribozyme can be engineered to basically any target site by just changing these few nucleotides in the binding arms to make them complementary to the target site. And in this way, we could even install M1A in natural tRNA sequences. So I should mention that this M1A is one of the key modifications that ensure proper folding of tRNA. When this is missing at one particular position, namely this position 9, then we do not get this a cloverleaf shape fold of the tRNA, but an extended hairpin-like structure, which is not non-functional. So this tiny little methyl group is really key to many tRNA functions. And now when we first make an unmodified tRNA, we can use the ribozyme to install the methyl group here at this particular position, or at these other conserved positions. We also tested in tRNA sequences from various organisms and could really show we have now the first protein-free tool in hand for site-specific RNA methylation. We also wanted to know how does the RNA actually do that? What is the structure and the catalytic mechanism of this ribozyme? And for this, we turned to crystallography, and we were indeed quite lucky that we got crystals that could be reproduced and that were also well diffracting. That's not something that is easily predictable. But in this case, we have a variable stem loop, and together with these two base paired regions here, we could engineer and screen many variants of constructs, and we found crystals only in the presence of the cofactor. And then, indeed, it was possible to solve the structure, and we found that during crystallization, the methyl group has been transferred. So in the final structure, in the active site, we find methylated RNA, and the demethylated guanine cofactor also is still bound. And what one can see here is how this guanine is held in place by hydrogen bonding with these two nucleotides. And this mimics how nature binds guanine in natural riboswitch sequences, a class of functional RNAs that bacteria use for regulating gene expression. They sense how much guanine is there, and depending on the guanine level, they either switch on or off a biosynthetic pathway. So now, by in vitro selection in the laboratory, we have found an active site that sort of mimics what nature has evolved over millions of years. But in addition to just binding the RNA as nature, binding the cofactor or the ligand as nature does, we have in addition now a chemical reaction catalyzed by this RNA. And the structure immediately suggests to us the chemical mechanism. So I showed you this pH rate profile, and this is very well consistent with this protonated cytidine here in the active site. So now with this understanding and further engineering of this ribozyme, we could even further accelerate this and can now, as you can see here, within a couple of minutes get full conversion, full methylation of a target RNA under very mild conditions in the absence of any protein. And so we can use this to install M1A, but we can also go back to our initial motivation, that is to develop tools for visualizing RNA. And now I just mentioned earlier that the SNAP tag was used as or is used as a very well-established tool for protein labeling. And those benzylguanines that carry the fluorophores, they are widely available. And we can now repurpose those with our ribozyme to site-specifically label RNA, to attach the chromophore at a particular position at a very defined RNA that is only selected by the base pairing arms here. And this expands our toolbox of, we have in the lab developed a series of other ribozymes that can target other nucleotides, other positions, namely the 2' hydroxy position, for example, by phosphodiester formation or this phosphonate ester formation. And now with this methyltransferase, we have one that can attach fluorophores or labels in general to the nucleobase, in this case, an adenosine. So what comes next? Well, I introduced earlier that there is 150 different modifications known. We have now one that can attach an alkyl group, a fluorophore, a methyl group to the N1 position of adenosine. But what about the many others? And eventually, we would like to use ribozymes for intracellular labeling of RNA. And yeah, this is the directions that we are going. There is much more that needs to be considered when we want to go into a cell. One question is, what would be a good cofactor for intracellular labeling? There, we also take advantage from our inspiration from the protein engineering field. Methyltransferases have been engineered to also transfer propargyl groups. This is a cofactor we made in our group that is modified at 2 additional sites that we then also used for in vitro selection. It's a SAM analog. SAM is the methyl group donor for methyltransferases. This we call now ProSetma for the propargyl group on a selenium center that carries an amide and an additional amino group here. And we use this for finding a ribozyme that can now, instead of the huge benzyl group or the chromophore attached to the benzyl group, transfer only this small propargyl group. And the ribozyme that came out of this study, we called Samurai for SAM analog utilizing ribozyme. This was spearheaded really by a postdoc in the lab, Takumi Okuda, who designed, synthesized the cofactor, carried out in vitro selection, and characterized the reaction product. And he also has the first data in hand that show that indeed a ribozyme works intracellularly. The advantage that he has that his cofactor is also cell permeable. So he can now engineer the RNA in the cell and show that the cofactor is taken up, used by the ribozyme, and the RNA gets alkylated and can then be fluorescently labeled by attachment of the fluorophore to this alkyne group. Yeah, so this is where we stand at this moment. We have a number of additional ribozymes in the pipeline. We think that eventually we will be able to use them as functional tags for covalent labeling of RNA in the cell to label and visualize, track the localizations, identify the interaction networks, and also modulate or potentially even rescue physiological RNA modification roles. There's way more to do in the future. What I showed you so far is that we found the first methyltransferase enzyme using this benzylated or methylated guanine inspired by DNA repair proteins, that we can also install bio-orthogonal functional groups like this propargyl group, that we can do this, achieve this with other bio-inspired cofactor analogs. And our next goals really are to address other positions in different nucleotides and to eventually modulate the cellular RNA modification landscape. So these were now only 2 projects out of the many that are going on in my lab in many different areas. I think I have already taken too much time, so I'm not telling you much more about this other very exciting area down here, where we have recently discovered a new chemical reaction for cross-linking of DNA. I jump over this quickly. We found a new 2+2 photocycle addition reaction that can connect two DNA strands. And this is now extremely useful for structural biology studies of DNA damage response and DNA repair pathways. And this is a very nice ongoing collaboration with the group of Carolina Kiska here at the University of Würzburg, who is really an expert in the DNA damage field and in the structural biology of those complexes. And with the formation of these new types of crosslinks, we really have now a means to trap these helicases in their active conformation and learn more about their functions. So overall, I think there is much to do in the field of RNA and DNA, and the modifications that nature offers and that we can develop in the laboratory. There is really many possibilities for future discoveries and for understanding biology in the first place, but also for developing new tools that allow us to study and manipulate if something is going wrong. With that, I would like to thank all my group members, those that are currently in the group, but also those that have contributed to many of the projects earlier. It's really a great honor to work with them, a pleasure to have motivated people in the lab who drive the projects and who are as excited about RNA chemistry as I am. Many of the projects would not have been possible without any of our collaborators here in Würzburg and those at other places, but especially in Göttingen. And also all the funding sources who support our work. And again, I would like to thank very much the Bayer Foundation for recognizing our work and also for today for giving us the opportunity to share some of our science. I stop now here. I'm open to discuss any of the other projects that you may be interested in, any additional questions about how I came to study RNA, DNA, other opportunities that we have here in Würzburg. Just let me know your interest. I thank you very much for your attention and the opportunity. And thanks everyone for joining.